Hi everyone, this is Ismaila Mariam and I will be speaking to you about the cerebellum, starting off with a bit of overview of its anatomy and its basic function, and then I will specifically focus on its intrinsic and afferent connections, which is basically its internal circuitry, and the afferent connections meaning its connections with the other structures of the CNS, such as the brainstem and the cerebral cortex. The cerebellum is a structure located at the back of the brainstem, and it is responsible for coordinating and regulating voluntary movements, balance, and posture. The cerebellum receives sensory information from the body and uses it to adjust and coordinate movements. It also receives information from other parts of the brain, such as the motor cortex from the cerebrum and the brainstem, which help to control and refine movement. Embryologically speaking, the cerebellum develops from rhombencephalon and specifically from the metencephalon, making it a part of the hindbrain. The cerebellum is divided into three components: the anterior, posterior, and flocculonodular lobes. These are three areas with different connections and different functions. The lobe that performs the most primitive function is the flocculonodular lobe, also known as the archicerebellum. Its main function is to control posture and balance. The second most primitively functioning lobe is the paleocerebellum, or the anterior lobe, and it is responsible for the tone of the muscles. The neocerebellum, also the posterior cerebellum, this is responsible for a more modern function, which is the coordination of movement. As you can see from the image on your left-hand side, the three areas are divided by certain fissures. So the primary fissure divides the anterior and posterior lobe, and the posterolateral fissure separates the posterior lobe from the flocculonodular lobe. On the right-hand side, you see an image of the posterior aspect of the cerebellum. This will allow us to visualize the functional areas of the cerebellum. In the middle, you see a longitudinal depression, which is called the vermis, and on the sides are the cerebellar hemispheres. The cerebellum controls balance and coordination ipsilaterally, in contrast to the cerebral cortex, which has signals going back and forth contralaterally. This means when you have lesions of the cerebellum on the left side, you will have problems with its functionality on the left side. The vermis, which is the structure connecting the two lateral hemispheres, has the body expressed on top of it topographically, meaning the vermis mainly has connections with axial musculature such as the neck, shoulder girdle, trunk, and hip girdle, and there's an area next to the vermis that is not mentioned in the slide called the paravermis, and in the paravermis, the topography is the presence connection with hands and feet. The cerebellar peduncles are a set of three bundles of nerves fibers that connect the cerebellum to the brainstem, and they are named based on their location and their direction of travel. Firstly, the middle cerebellar peduncle. This is the largest of the three peduncles in terms of the number of fibers it has. It connects the cerebellum to the pons, and it carries input fibers from the cerebral cortex to the cerebellum. And it includes information about planned movements and ongoing motor activity. Secondly, the inferior cerebellar peduncle. This is the smallest of the three peduncles, and it connects the cerebellum to the medulla oblongata, which is the lowest part of the brainstem. It carries input and output fibers, carrying information about balance, proprioception, and other sensory feedbacks from the body and the vestibular system. And lastly, the superior cerebellar peduncle. This is the largest and most dorsal of the three peduncles. It carries output fibers from the cerebellum to the thalamus and the midbrain, and it also receives input from the cerebral cortex and other parts of the brain. The internal circuitry is located in the cerebellar cortex, which is the outer layer of the cerebellum. It is organized into several distinct layers of cells, each with a specific role in coordinating movement and regulating posture. The circuitry has the granular cell layer, Purkinje cell layer, molecular cell layer, and the deep cerebellar nuclei. The circuitry of the cerebellum allows for the integration of sensory and motor information, and eventually the precise control of movement. The granule cells and Purkinje cells specifically play a key role in processing and transmitting the information that it receives, while the deep cerebellar nuclei acts as the final output pathway from the cerebellum to the other parts of the brain. There are two regions of gray matter in the cerebellum, the cerebellar cortex and the deep cerebellar nuclei. The cerebellar cortex is located outwardly on the cerebellum, whereas the deep cerebellar nuclei are deep within the cerebellum, and in between these two layers of gray matter is the white matter. As mentioned in the previous slide, the cerebellar cortex is made up of three layers: the superficial molecular, the middle Purkinje cell layer, and the deep granular cell layer. I will go into the details of these in the next slides. Granular cell layer consists primarily of two cell types, granule cells and Golgi cells. The granule cells are excitatory cells, and they have glutamate as their key neurotransmitter. And their axons are parallel fibers that stimulate Purkinje cells. What that means is, as per the diagram, you can see that the cell in blue is the granule cell. It has its nuclei in the granule cell layer, whereas its axon, its dendrite, is extending upwards into the molecular cell layer, bifurcating and running along that layer, synapsing with the Purkinje cell here in red. The other cell type is the Golgi cell. These are inhibitory cortical interneurons. They control the internal circuit by inhibiting certain signals between the circuit. And glycine is the key neurotransmitter. Its axon terminate on dendrites of granule cells. The Purkinje cell layer is located beneath the molecular layer, and it contains large, flat Purkinje cells. These neurons are the only output neurons of the cerebellar cortex, and they receive input from granule cells and other sources such as the climbing fibers and the mossy fibers that I will talk about in the next slide. The Purkinje cells send their axons to the deep cerebellar nuclei, and they are inhibitory, and they have GABA as their key neurotransmitter. The molecular layer is interesting. It is the outermost layer, and it contains a dense network of interconnecting fibers, including dendrites of Purkinje cells, axons of granule cells, and various types of interneurons, such as stellate and basket cells. We know that there are many fibers entering the cerebellum, from the spinal cord, from the cerebral cortex, from the brainstem. The fibers that are entering the cerebellum are categorized into two kinds. The fibers that are entering from the inferior olivary nucleus and the rest. The fibers that are originating from the inferior olivary nucleus and entering the cerebellum are called climbing fibers, as these fibers are climbing up until the Purkinje cell layer as they enter the cerebellar cortex, and the rest of the fibers coming from multiple locations such as the cerebral cortex, the spinal cord, they are referred to as mossy fibers. And remember that all fibers that enter into the cerebellum are excitatory. Now let's break down the circuit. The cerebellar nuclei, which is the structure in pink, is present not in the cerebral cortex but rather deep within the cerebellum. It is one of the two areas of gray matter of the cerebellum. As you can see, the climbing and mossy fibers in blue and climbing fibers in purple, these are fibers that are, as I mentioned previously, entering into the cerebellum. We know that these are excitatory, so they are going to synapse with the cerebellar nuclei and they're going to release excitatory neurotransmitter. The climbing fibers are specifically going to release aspartate, and mossy fibers are going to release glutamate. The climbing fibers, as you can see, are further going towards the cerebral cortex and synapsing with the Purkinje cell. The Purkinje cell, which here is in orange, has its nuclei in the Purkinje cell layer and its dendrite extending into the molecular cell layer and its dendrite extending into the white matter synapsing with the cerebellar nuclei. The Purkinje cell is inhibitory as opposed to the fibers climbing and mossy. So since you can see that the climbing fibers are excitatory, they will excite the cerebellar nuclei and they will also excite the Purkinje cell. The excitation of the Purkinje cell will eventually result in the inhibition of the cerebellar nuclei, as they will release the neurotransmitter GABA. So this means that the climbing fibers are also stimulating the cerebellar nuclei, but they're indirectly inhibiting the cerebellar nuclei as well. Let's talk about the mossy fibers. The mossy fibers here in blue, as you can see, are also synapsing with the granule cells. The granule cells are cells present in the granular layer whose dendrites are extending onto the molecular layer. Their axons eventually bifurcate, forming long strands in the molecular layer. And as you can see, they're synapsing with multiple kinds of neurons, especially with the Purkinje cells. So these long strands are going to synapse with millions and millions of Purkinje cells. And so since mossy fibers are synapsing with many granule cells, and these granule cells are synapsing with many Purkinje cells, this allows one single mossy fiber to have a very diffuse effect. As we know, Purkinje cell eventually inhibits the deep cerebellar nuclei. That is what it will eventually result in. There are certain inhibitory neurons in the internal circuit that are responsible for controlling the circuit. The Golgi cell, which is synapsing with in the granular layer with the mossy fibers, as well as the granule cell. And they're also synapsing with the parallel fibers from the granule cell in the molecular layer. So how does it work? It basically receives either stimulation from the parallel fibers in the molecular layer, and then it's going to inhibit the granule cells in the granular layer. This is basically an auto-inhibitory function. Or it could receive stimulation from the mossy fibers, and that will then cause it to inhibit, send an inhibitory signal to the granule cell layer. There are two other inhibitory cells, the stellate cells and the basket cells. Now, what are their functions? As you can see in the picture, they are synapsing with both the Purkinje cell and the granule cell, parallel fibers. So what happens is the granule cell, as you can see, via the parallel fibers are synapsing with the Purkinje cells and stimulating the Purkinje cells. Now imagine there are multiple Purkinje cells in this diagram. So it's not just one Purkinje cell stimulated, it's millions of them. So the granule cells with the parallel fibers are also synapsing with the stellate cell and the basket cell. These two are inhibitory neurons. They are going to inhibit Purkinje cells. So they have synapses with some Purkinje cells and not with others. This means that the granule cell is indirectly inhibiting some Purkinje cells as well. This allows for some Purkinje cells to be activated while others to be inhibited. What this will result is in the sharpening and enhancement of the signals of the Purkinje cells that are left activated. So in summary, the granule cell is indirectly inhibiting the Purkinje cell via the stellate cell and the basket cell. Let's talk efferent connections. These are basically pathways coming from the cerebellum to the other structures. They are originating from the cerebellar nuclei, as we talked about in the internal circuitry slide, and they travel mainly via the superior cerebellar peduncle. And the cerebellum forms connections with the thalamus, the vestibular nuclei, the red nucleus, and the reticular formation. And they form the following tracts: cerebellovestibular tract, cerebello-reticular tract, and etc. These efferent pathways of the cerebellum are responsible for modulating and fine-tuning motor output to achieve a smooth, coordinated movement. Dysfunction of these pathways are going to result in movement disorders such as ataxia, dysmetria, and dysarthria. The cerebellovestibular pathway, it originates from the flocculonodular lobe of the cerebellum, and it projects to the vestibular nuclei in the brainstem. Its main function is to control balance and posture. The cerebellothalamic tract is a major afferent pathway which stems from the deep cerebellar nuclei, projects onto the thalamus, which in turn projects it to the cerebral cortex. It is also known as the dentatothalamic pathway because it mainly stems from the dentate nucleus. This pathway is responsible for fine-tuned motor output to achieve smooth, coordinated movements, but it also contributes to cognitive processes such as planning, decision-making, and attention. Then you can see the cerebello-rubral tract. This is a connection from the deep cerebellar nuclei, mainly the interposed nucleus, to the red nucleus that is present in the midbrain. The red nucleus, in turn, sends axonal projections to the spinal cord, and it is mainly responsible for controlling motor functions of the body by controlling muscle tone, posture, and also coordination of movement. The cerebello-rubral tract also helps to maintain balance during movements, and it contributes to the regulation of eye movements. Here are the references that I used, and I hope you found this video helpful.